Okay, it is now 12:38, and I am very pleased to introduce our first speaker, Andrea Guedes, in the Department of Chemistry and Biochemistry, who will be talking to us about genetic selection of sarcosine-specific synthetic riboswitches from a glycine riboswitch. And you have 5 minutes.
Thank you so much for this opportunity to talk about my work. I'm going to talk about the genetic selection of sarcosine riboswitch, as you say so. And first, I want to define what a riboswitch is. A riboswitch is a biological switch that can regulate further processes. Basically, it's composed by two parts: a tamer region, which works like a sensing area, and expression platform. You can visualize it like a plug and a wall outlet analogy. Basically, the molecule, or in the case of the example, the plug is going to bind to the aptamer region or the wall outlet, according with my example, and it's going to cause a response in the genes that are close to that area. This is made from RNA. So we can use this technology to activate or deactivate specific processes. And we decided to use this technology to create a biosensing system that allows us to check for the concentration of specific metabolites in body samples. So we propose to use the glycine riboswitch, which is a riboswitch that is responsive to an amino acid that is called glycine. And basically it's a turn-on riboswitch, which means that detects this substance and activates a further process. And we decided to use it to create a riboswitch specific for sarcosine. Sarcosine is a small molecule that is present in the body, but it can be in higher concentrations in patients that have prostate cancer or that have poor prognosis in cancer cases. So we want to be able to create a system that can sense the concentration of this molecule, and it can give us a signal so we can know how much of that is present in a body sample. What we did is we took the glycine riboswitch and we modified the sensing area to create a whole library that was further tested for the presence of sarcosine. We decided to randomize this and generate a library that has billions of variants. And then that library was further tested. The way that it was tested was by attaching a dual genetic screening or a dual gene that encodes two specific proteins. So what we want is to see if the riboswitch is only responsible to sarcosine. That's our goal. So we had a first positive selection in which we prepare all our growth media in presence of sarcosine and all the nutrients that the cells need to have to grow. We introduced the riboswitch in the cells, and then we added chloramphenicol. So if the riboswitch is turned on in presence of any molecule that is in our plates, it's going to activate the presence of an enzyme that is going to modify this antibiotic, and in other words, the cells are going to grow. But because there are other things present in that specific plate, we want to make sure that the riboswitch only responds to the concentration of sarcosine and nothing else, because we only want to sense sarcosine. So for that, we incorporate a negative selection in which we have a substance that is called 5-fluorouracil, and we don't have sarcosine. When the riboswitch is turned on and this substance is present, it's going to cause the production of a gene that is toxic. And in other words, the cells are going to die. And only the riboswitch that wasn't activated by sarcosine is the one that is going to make the cells to grow. And we are going to have or positive signal. So we repeat this several times, and then we retest the clones to make sure that they only respond to the concentration of sarcosine. And we can see it here, how we have cells in presence of sarcosine, and we don't have any cells when we don't have the sarcosine and we have higher concentrations of our antibiotic. And because we want to have a sensing system, we want to incorporate some kind of signal. And in this case, we incorporate a gene that is called a marker gene that expresses an enzyme that basically acts like a scissor, and it cuts this molecule, generating a yellow color. So the more intense the color is, we have more expression of, or that riboswitch is more activated in the presence of a sarcosine. So I can show you here, this is the signal that we have. That's basically how intense the yellow color is. And here's the concentration of our metabolite. So we wanted to increase as the concentration of sarcosine increased, so it can act like a sensor. And we were able to identify two clones, the orange one and a green, this olive one that also increases as the concentration increase. So in conclusion, we were able to change the specificity of these ribose switches towards small molecules, and we're gonna keep testing this library for another molecules to be detected. And I want to thank Dr. Yu for all his mentorship and the College of Science and Engineering for all their funds. And I will be happy to answer any questions that you have. Thank you.
Okay. Excellent talk. Does anybody have any questions for Andrea? All right, we have a question from Ken Richardson. To what extent has this riboswitch technique been used before?
So there are some cases of RNA-based sensors that are still in vitro analysis. So basically the cells have been cultured with these ribose switches, and it has been studied the concentration dependency relationship between them. It's not still used as a biosensor, but the technology is being created and it's being tested.
All right, and we have another question from Laura Luque saying, great presentation.
Thanks.
You have a proof of principle. How would this be translated to be used in humans?
Yeah. So basically, the next step will be to design a proof in which we can have either a cell-based system in which we can have maybe like a chip or something in which we can have that kind of systems, and then like adapt it to a detector. So we can kind of translate that signal that we get based on the intensity of color in numbers with a calibration curve, and then like we can translate that to the real numbers.
All right, and then we have a question from Bomi. Is it used for detecting cancer?
No, so it can detect, we want to use it to detect molecules that can be misregulated in body. So basically with a body sample, you can like detect if that molecule is in excess, and we can, and there are research that are related with the existence of excess of that compound in patients that have cancer, specifically prostate cancer. So it can be related, and it's also characterized as a biomarker.
All right. Thank you very much.
Thank you.
Are we doing a Zoom clap between speakers? Or okay, so our next speaker is Ali Benson. Okay.
Sorry, I'm promoting Ali to co-host right now, so she can share.
Will do, and I'll do her her introduction. Our next presenter is Ali Benson, who will be talking about employment decisions in mothers of children with autism spectrum disorders. You're
yeah, I unmuted you, but it's not showing me the option to make you co-host right now. Sorry.
We allow multiple participants to share on the screen. Will that work? That works sometimes as well. All right, so that is enabled.
Okay, I just got it to share. Okay, so my name is Ali, and my research is on employment decisions in mothers of children with autism spectrum disorder, or ASD. So parents of children with ASD are 4 times as likely to quit, change, or not take a job compared to to parents with typically developing children. And it is the mothers who are disproportionately quitting their jobs compared to fathers to care for their child with ASD. Further, research suggests that mothers of children with ASD experience more stress and lower overall health ratings than both parents of typically developing children and children with other special needs. And so in light of this prior research, I asked two questions. First, what are the factors underlying the reasons mothers of children with ASD decide to quit their job versus continuing work outside the home? And second, what are the effects of quitting a job to care for a child with ASD on a mother's mental health versus the effects of staying in a career while parenting a child with ASD? My fire alarm is going off. Just 1 second, it might stop.
You should be unmuted.
Okay, I'm really sorry about that. I know my time has been cut, so I will just abbreviate some. So we had 24 mothers who quit their job to care for their child with ASD, and 43 mothers who remain in the professional workforce while raising their child with ASD, who completed our survey. That was an online Qualtrics survey that included mental health measures, demographic measures, and questions about employment decisions. And so our results, we found that working mothers derive more meaning in life than mothers who quit their job to care for their child with ASD. And so a lot of the working mothers responded that some of the reasons they decided to remain in the professional workforce was because they found their careers rewarding and also because they wanted to retain independence outside of motherhood. Additionally, we found that stay-at-home mothers reported feelings of not accomplishing anything at home. So those responses kind of illustrates the results we found there. Additionally, mothers who quit their job experience more depressive symptoms than working mothers. So a lot of these stay-at-home moms reported feelings of loneliness and isolation, and also not having an outlet to relieve their stress. And finally, they reported things like not getting enough adult interaction. And so those responses kind of illustrate the results that we did find there. Finally, we found that working mothers experience higher levels of state anxiety and perceived stress than mothers who quit their job to care for their child with ASD. So a lot of the working moms reported a lack of flexibility in their careers, giving them stress, and also stress from balancing work and home life. And so as you can see, there's kind of a trade-off here. So the working mothers are experiencing higher levels of state anxiety and perceived stress, but on the other hand, they do derive more meaning in their lives than the mothers who quit their job to care for their child with ASD. And so finally, we found that the biggest factor in a mom's decision to remain in the professional workforce was financial needs, whereas the biggest factor for a mom's decision to quit her job to care for her child with ASD was a child's needs. And there was no statistically significant association between mother employment and job prestige when deciding whether to leave or stay in her job. So the limitations of this study were that we had difficulty recruiting mothers who quit their job to care for their child with ASD. And the majority of our participants were white and individuals above the poverty line. And finally, I would advocate for increased flexibility in leave time to relieve some of the state anxiety and perceived stress of the working moms. I would advocate for better support groups to alleviate some of the depressive symptoms of the stay-at-home mothers. And finally, I would advocate for more respite care to help all of these mothers of children with autism spectrum disorder. And I do apologize that my fire alarm went off, but now I would like to take some questions.
All right. Does anyone have any questions? Excuse me for that's my timer going off for 5 minutes. All right. So we have a question from Laura Luque. Great adjustment and didn't take anything away from your presentation. And I would like to echo that. You handled that with absolute aplomb. What kind of support will mothers need at their job place to keep their job while still feeling that the needs of their children are being addressed?
Yeah, so that's a great question. A lot of them reported flexibility, needing more flexibility in leave time. And so it's common for children with ASD to have a meltdown during the day. And sometimes the parents are the only people who can alleviate that. And so just more flexibility in leave time from employers would really help both the moms and their stressors. And I believe it would help the children with ASD as well.
All right, and we have a question from Becky Biddle saying you were so gracious under adverse situations. Did you look at the severity of the condition versus the mother's decisions?
A little bit. I will say that this study was skewed towards people who are higher functioning with ASD. So I would say more often than not, the moms in this study had children who had high functioning ASD as opposed to low functioning ASD.
All right. Thank you very much. I think we need to, if there are no more questions, I think we need to move on. Okay. Our next presenter is Jim Herendeen. I'm sorry.
Hello, yes, that is correct.
All right, excellent. You should be co-host. You can share your screen. And he will be talking, he's working with Robert Biddle, and he will be talking about a continuous feed and return system for a rotary dryer.
Is it possible to have another participant share their screen, Brandon Beatty?
Okay.
Okay, just this slide will begin. All right, so for our project, we are designing a closed loop continuous feed and return system. And the participants in this project are Brandon Beatty, Drew Peniter, Eugenia Scala, Jim Herendeen, and Haley Mallion. So as an overview, our client tasked us with designing a closed loop continuous process for mined limestone for the purpose of modeling the lifter characteristics inside the dryer to increase drying time efficiency. The most important specifications from our client included that this system be a continuous process, attain a material throughput of 20 tons per hour, and integrating used client equipment. The most challenging aspect of this project was designing individual components, including the hopper and base structure, integrating an existing feeder, designing a short screw base to hold a material catch and a screw conveyor, and designing 2 screw conveyors. Brandon, I think you're muted. Well, as far as the hopper and base goes,
yeah, I can
go
talk
ahead.
now. I'm sorry for the delay. I was not able to unmute myself. So the first couple of components include the hopper and the hopper base. We were tasked with designing a reservoir to funnel material into the feeder, otherwise known as a hopper, with a capacity of 10 cubic feet. This volume was established based upon the total amount of material that could be in the dryer, short screw, and long screw at one time. The specific design of the hopper depends on the feeder we are using to feed the material into the dryer, which is a pan feeder. Therefore, the ideal hopper design, which is shown in the middle figure on your screen, was used for our design. This design depends on many parameters, but the main components are the gate height G and the throat T. These dimensions are proportional to the largest particle size that will be used in the continuous system, which is 4 mesh. Finally, the base needed to be built such that it supports the loads of the material in the hopper itself, along with the feeder. One of the major challenges was finding the correct height of the base such that the long screw, such that the angle of the long screw feeding the hopper did not exceed 45 degrees, but the base was high enough to feed the material into the dryer at the correct height.
Eugenia, you might be muted. As for the feeder, this was a piece of equipment provided by the client that we needed to integrate into the system. Because of the height of the cylinder being 6, or the middle of the centroid of the cylinder being 6 feet, and being 6 feet, the feeder needed to be placed at a discharge height a little bit under 6 feet. So in order to ensure this, the feeder will be supported by steel cables attached to the front and rear assembly of the base structure. We needed to reconfigure the feeder in order to design a change from the trough from a tubular feeder into a pan feeder to achieve the desired feed rate of 20 tons per hour. And now the short screw base. Haley, you might be muted. But as far as the short screw base, we needed the short screw base to be able to hold a material catch at the discharge of the cylinder, as well as a short screw that would continue to move the material through the system. The material catch itself had 2 outlets, one going to the short screw conveyor and one going to deplete the system of all the dried material, and a piece that covers each outlet, allowing for the user to choose which outlet they want to use. With the help of some clamps, and the intention of this base was to hold both the material catch and the short screw in a fixed position to ensure a continuous process.
All right, so I'm going to talk to you guys about the short screw. So we had 2 screw conveyors used in this, a short screw and a long screw. And the important client spec for us was the need to maintain a throughput of 20 tons per hour. So this was kept in mind as we were looking at options for these. And so the short screw specs primarily came from the client's existing on-site screw. So using an effective length of 60 inches at an angle of 30 degrees, we used their 2 horsepower motor, and we found that this was enough to achieve the specs of 20 tons per hour, but that we would need a variable frequency drive attached to the motor so that we can adjust the feed rate to make this process move along a lot smoother. And for the long screw, it was a complete rebuild, as they had an existing on-site long screw, but it was rusted and needed to be replaced. So we had it redesigned. We outsourced it to an effective length of 174 inches at a 45-degree incline to return material back from the hopper to the discharge. And so the long screw itself will also need a VFD so we can vary the frequency to the feeder. And with both of these screw conveyors, primary process for which we can return the product back to the hopper.
That's time, guys. So we have time for one question. So we have something from Becky Biddle. Jim saying to Jim, good job to just keeping this going. Do you have any concerns with impacts of rocks or parts of general fatigue or on the parts?
The main part that we would have, or main component of the system that we would have a challenge with during the design of this, was actually these screw conveyors. Initially, we wanted to use like a conveyor system, like you might think of like a conveyor belt in, like an airport or something, but due to the abrasive nature of limestone, that is why we had to use a screw conveyor. And that is the part that we're most worried about, like as for our wear and tear. In addition to the fact that this is only being run for about, maybe 2 hours on a day, and so it does need to be continuous in the system, but it's not being run 24/7. So no, that is something that we considered in this design.
Okay, I think we've got probably actually time for about one more question because we're running a bit ahead of schedule, overall ahead of schedule right now. So I will take host privileges and ask, what
I
were,
have this on mute, right? I can't talk. That's what if
I think you have.
pardon?
I think that was a mistake.
Okay, well, what I wanted to know is what exact specifications were you given from the client?
The major parameters would be having a throughput for the system of 20 tons per hour. And initially, that first value was 36 tons per hour, but due to the scaling and size of our system, we were able to, effectively convince our client, Loas, that was an unrealistic expectation for the throughput of a system. So that was basically the biggest design for parameter for the closed loop system itself, is just to ensure a continuous process at that high rate of speed.
Okay, all right, let's move on. Our next presenter is Matthew Espinoza, who is going to be telling us about you are what you are willing to eat, willingness to try new foods, impacts perceptions of sexual unrestrictedness and desirability. All right.
Thank you. So to begin, I'd like everyone to try and imagine that they're out on a first date and you and this potential dating partner are going to go out to have some dinner at a nice restaurant. And when it comes time to order what to eat, you have to make a decision between having some boring old regular cheeseburger that you've had a million times before, or trying something new and unfamiliar to you, like perhaps an exotic plate of sashimi. And while you may think that this choice in food would have no impact on whether or not your potential dating partner would want to have a second date with you, a recent survey of over 4,000 single men and women found that the majority of people find a reluctance to try new foods to be a major turnoff. Because you see, how and what you eat changes how other people see you. So, for example, people that eat smaller meals with more fruits and vegetables and are just generally a lot healthier in terms of their diets, these people tend to be seen as more physically attractive. Whereas on the flip side, people with dietary restrictions, such as those with gluten allergies, tend to be seen as more complaining and judgmental. And so what we were interested in looking at is whether or not people make mating relevant inferences based off of a potential dating partner's willingness or reluctance to try new foods. And so to do this, we conducted 4 studies, though I will be focusing on studies 2 and study 4 here today. And so in our second study, we recruited a sample of undergraduate college men and women and showed them 2 online dating profiles that resembled profiles you could find on popular websites such as OkCupid. And while these dating profiles were similar, they were different in that they described people that were different in terms of their willingness to try new foods. That is, one of the profiles described someone that was very willing to try new foods, whereas the other profile described someone that was very reluctant to try new foods. And after viewing each of these profiles, participants then rated the potential dating partners that were described. They rated them on various different mating relevant characteristics, such as their physical attractiveness and their romantic desirability. And so what we found here, as you can see in that box on the top right-hand side in the graph, is that compared to potential partners that were very reluctant to try new foods, participants rated the willing to try new food target as being much more sexually unrestricted. That is, they thought that someone who was willing to try new foods was also someone who would be very willing to have a short-term uncommitted sexual relationship. And so moving on towards our 4th study, here we wanted to look at whether or not this relationship that we found in study 2 was mediated or explained by differences or changes in perceptions of the potential dating partner's sexual disgust sensitivity. And so once again, we collected a sample of undergraduate college men and women and showed them the same 2 online profiles from study 2, Only this time, in addition to the other mating relevant characteristics, we also had participants rate these potential partners and with regards to the potential partner's sexual disgust sensitivity. And so what we found here, as you can see in the figure on the right, is that when people saw that a potential dating partner was very willing to try new foods, they then assumed that this person would also have a reduced level of sexual disgust. That is, they saw this person as being someone who would be very unlikely to get grossed out during sexual situations or bisexual behaviors. And it was in fact this assumption, this change in perceived sexual disgust that then led people to make these inferences about the potential partner's unrestricted sexual behaviors. And so to summarize, basically our results provide evidence that people, at least subconsciously, are aware of the connection between a potential dating partner's willingness to try new foods and their other sexual behaviors. And that seeing information about a potential partner's willingness to try new foods leads people to make assumptions about that potential partner's sexual disgust, as well as their willingness to engage in short-term uncommitted sexual relationships. And these changes in perceptions likely influence how people make decisions about who they want to pursue as a romantic partner and who they'd rather keep as a friend. So the next time you're out to dinner on a first date, maybe try ordering something you never had before, because it might just help you secure that second date. Thank you.
Thank you very much. Do we have any questions? This is from Gary Boehm. Could there be an interaction between willingness to try new foods and their attractiveness, in which this unwillingness was viewed more favorably in attractive people who could afford to be choosy, but not an unattractive who couldn't?
Yeah, there could be, in fact, an interaction here where it could, if someone who's very attractive is also very willing to try new foods, that might make them even more attractive to someone based on what type of partner they're trying to pursue. Whereas someone who's, perhaps not the most attractive person in the world, because we want to be nice and not call people ugly, if they're very willing to try new foods, that might make them a little more attractive. So yes, there could be an interaction there.
Okay. Any other questions? All right. Thank you very much. Okay. All right. Our next speaker is Megan Alexander, who will be talking to us about assessing lake sediment for use as an amendment. And you should be able to share your screen.
Can you hear me?
Yes, we can. All right.
Okay, so hello, my name is Megan Alexander, and I will be presenting my group's SRS project for assessing lake sediment for use as a soil amendment. And so we can start off by talking about how agricultural soils require the presence of 3 basic plant nutrients, which are nitrogen, phosphorus, and potassium. Each nutrient has its role in plant growth, and their deficiencies have adverse effects. So therefore, a detailed analysis was required to analyze whether soils from a lake were sufficient in terms of these nutrients. And also if they could be used as an agricultural soil amendment on a particular plot of land. And so in this case, the plot of land was farmland, and we were looking at a soil amendment that was good for growth of lettuce and tomatoes specifically. And so experimental study by way of specific chemical analysis methods, LOI or loss on ignition, spectroscopy, was used to look at water content, pH, EC, which is electrical conductivity, and soil organic carbon. And so phosphorus and nitrogen content in the lake sediments these values were used to determine what contents and nutrients in the lake would need to be optimized for crops of interest. And so the results from our experimental procedures allow the property owner to effectively make use of their resources. And so specifically, we looked at nitrogen versus phosphorus after taking soil organic carbon values using spectroscopy. And we found that the values from spectroscopy were much more accurate in regards to soil organic carbon. And so looking at nitrogen versus phosphorus on the site, this is before amendment, we optimized for phosphorus because we found that nitrogen levels across the board were already steady in terms of what would need to be optimized. Phosphorus, on the other hand, was lacking. And so we chose to optimize for phosphorus due to its low spread. And so this is what we calculated as application of soil amendment. The first graph you see here shows phosphorus content of the site, and the darker areas are where we would apply larger amounts in milligrams per kilogram of phosphorus to the site itself. And the middle graph shows the weight that we would add. Again, the darker areas show pounds per acre of where we would add phosphorus content to the site. And then the third graph shows amendment depth required for the site. And so, as you can see, weight and amendment depth are almost exactly identical in terms of what we would add to the site itself. And so we looked at water content. And so water content affects availability of water for plants and soil erosion. We had a very high water content, on average 44%. And so this increases soil moisture, which also determined whether or not we would use phosphorus or nitrogen as a soil amendment. As well as electrical conductivity, it determines the salinity of the soil and is used as an indicator for soil health. And so the ideal EC range for agricultural soils would be between 200 and 1,200. And so we found that our soil itself was within this range once optimized for phosphorus. And so in conclusion, our soil amendment was found to have high water content, ideal pH conditions for specific crop productivity, tomato, lettuce, onions, ideal SOC values, low nitrogen values, and high levels of phosphorus. So testing for more variables would increase samples from the amendment. It would avoid human error during the analytical process and improve measurement and sampling techniques. And so in the future, we would like to conduct more trials in terms of chemical analysis, and we could possibly use elemental analysis, ICP, and TGA in order to make our study more accurate and precise in terms of what we would use as an amendment for this crop. So thank you.
All right. Thank you, Megan. Does anybody have any questions? Okay, are there any of these additional techniques that you discussed at the end here? What do you think your next step is?
So we did take on this project from a recent group who studied something similar on the same cropland. And so maybe using the values and the data that they have found as well and combining it with our own to see like the differences between what we found and how to move forward from there. As I said before, for the future, testing for more variables and conducting more trials would help to more accurately portray our data in the future.
Okay. All right. Okay, excellent. Thank you.
Thank you very much.
All right. Okay. All right. Our next speaker is Axel Saborin.
Yep.
All right. Why don't you share your screen for us? Okay, and he will be telling us about preparation of clickable monomers compatible with automated PNA synthesis.
Yeah. Can you hear me? Okay, perfect. So, hi everybody, and welcome to my presentation where we'll be talking about the preparation of a clickable monomer compatible with automated PNA synthesis. So peptide nucleic acids, also called PNAs, what are they? First, we need to talk about DNA. So the general structure of DNA is composed of a phosphate sugar backbone in black here, onto which are attached various nucleic bases, so adenine, cytosine, guanine, and thymine. And so a PNA is basically the same thing, those nucleic bases, thymine, guanine, cytosine, adenine, linked together by a peptidic backbone in red here instead of the phosphate sugar backbone. And so they can be made from like very small units here through peptidic oligomerization. And the first report of a PNA in science or chemistry has been done by Peter Nielsen in 1991. So what is very interesting about those PNAs is that they are very stable due to the neutral peptidic backbone. They're easy to synthesize, as I said earlier, with peptidic coupling. They're very easy to derivatize, as you can see here. For example, in the past decade, there are 3 examples of differently functionalized monomers. And also, they have a very high affinity with DNA and RNA strands. And because of that, people have found applications in the medical field, such as antisense therapy drugs, gene editing tools, or fluorogenic probes. However, like everything, there are limitations to their use. And the main limitation of PNAs is their solubility in vivo, which is also due to their neutral backbone. And because of that, there is also issues with the intracellular delivery, which is a problem since that's where we want the PNA to go, since that's where the DNA and RNA are located. And there is also the issue of a rapid clearance. So that's why people are designing new monomers with different functional groups to try to prevent and circumvent those issues. And that's why our group designed a new monomer represented here with a propargyl amine moiety here and the diamine here. So it would lead to that type of PNA. And the propargyl amine is very interesting because it acts as a handle for functionalization post or pre-oligomerization. So if we want to have the red functional group, blue and green to be identical, we functionalize after the oligomerization. But if we want different functional groups, we can do it pre-oligomerizations. And it's very interesting because we can attach a variety of different groups, such as charged groups represented here, which would increase the solubility of the PNA, or for example, a fluorescent tag, which would be like used for like the probes, or simply a delivery vehicle if you want to make sure it reaches its target. So now I'm going to present the synthesis. So we started from aminoacetal represented here that was reacted with propargyl bromide to yield the secondary amine in an 84% yield, which was protected with a smoke in a very high yield. The acetal was then deprotected with the aldehyde into the aldehyde, also in a very high yield, which was then reacted with the glycine derivative here through a reductive elimination to give this corresponding this intermediate, which we have not isolated but engaged directly in the peptidic coupling with the 2 carboxylic acids that gave this monomer here. So one with the thymine bases in 48% yield, and with the CBZ protected cytosine, so 2 DNA bases in a 37% yield. So it's very interesting because we've been able to make those 2 monomers on gram scale synthesis. So that's a bunch of, or like a lot of product to study. It's very short, 5 steps with pretty good overall yields, 37% and 28%. And very important, the the compounds are bench stable like over months. So once we've done those monomers, the next thing to study was the selective deprotections of the two groups. So the therapeutic group here and the FMOG group here. So we were able to deprotect the therapeutic group in presence of TFA in a quantitative yield in the case of the thymine monomer. We were also able to deprotect selectively the FMOG in presence of piperidine in the case of the thymine with 84% yield, or the CBZ protected cytosine and 85% yield. So those are like very good results, because we can make grams of the compounds. They are like very stable, and we can selectively deprotect them. So do whatever we want with them in a chemical sense. So now what's left is the synthesis of the remaining DNA RNA basis monomer, some examples of click chemistry to functionalize them, and finally finding collaborations to test our monomers. So thank you very much. And I also would like to thank Dr. Jean-Luc Monchamp and Grace Inuel that worked very hard with me on that project.
Thank you very much. Does anybody have any questions? All right, this is from Karen. Can you explain the concept of click chemistry for us?
Yes. So here, so the click chemistry is basically using that triple bond, triple carbon-carbon bond here, and reacting it with, you can see here, the 3 nitrogens. And so basically, I don't see if you can see me, it just comes as on top of each other, and they click together to form this triazole ring, so the 5-member ring with 3 nitrogen and 2 carbons. So the 2 carbons here come from the alkyne represented here.
Okay, and we have a question from Kayla Green. The deep protection methods you used are most familiar for me for peptide synthesis. How hard is the work up for this in solution? Specifically, and I'm gonna pronounce this wrong, I'm sorry, the piperidine deprotection.
So, well, for the TFA dichloromethane, like the deprotection of the therapeutic ester, it's very easy. Like you mix them together, like you let them react until there is no longer any starting material, and then you just remove all the solvent and reagents, and your product is a nice solid that is remains in the round bottom flask. For the piperidine deprotection, we are also able to remove the DMF and the piperidine, and we just run a quick silica gel chromatography column on it, and it's more like a filtration on silica actually, and it gives the product pure as a crystalline solid also. But I guess we could also try some recrystallization.
All right, thank you again. All right, our next speaker is Monica Parra, who is going to be talking to us about the effects of trait imaginability and extrapolation on metacognitive outcomes.
Okay, hello. So I'm Monica, and again, this project will be looking at the effects of trait imageability on extrapolation and metacognitive outcomes. So to introduce this, past research has shown that people can adopt more extreme attitudes, or in other words, become self-radicalized towards a group simply by thinking about them. A number of studies in our research lab have found that people can become self-radicalized towards outgroups when they extrapolate beyond the negative traits that were presented to them about the outgroup. So for instance, sometimes we will tell participants that a certain group has exhibited a number of negative traits, and then we will ask them to rate the likelihood of how they would perform an additional number of negative traits. And in a recent study, we manipulated whether it was easy or difficult to form a mental image of certain traits during an extrapolation task, and found the participants that extrapolated to traits that were more difficult to form a mental image of reported more self-radicalization compared to those that had traits that were more relatively easy to form a mental image of. And the current study was interested in whether these effects of trait imageability on extrapolation might impact how much people think they know about an outgroup. So for our method, before participants started, we had them read a description about people from a fictitious migrant caravan, and they were told that US citizens had lived with these people at the time, and they described the people as being suspicious, impulsive, manipulative, etc., just to name a few traits. And then participants were assigned to one of three conditions. So there was the rehearsal condition, where we had them retype the traits that they were originally given about the group. Then we had the difficult to image, or the low imageability condition, where participants rated how likely it was that the group would exhibit 11 moderately positive traits and 11 moderately negative traits. And again, these were rated as relatively difficult to form a mental image of. And then we had the easy to image, or the high imageability condition, where participants also rated how likely it was that the group would have 11 moderately positive and 11 moderately negative traits, and these were relatively easy to form a mental image of. All participants were then reminded before they started the main survey that they knew nothing about how the people from Ossetia would behave, and they were then asked to rate how much they feel they know about the outgroup on a scale of 0 to 9, how confident they were that what they knew about the outgroup was to be true, and lastly, how accurate they thought that the US citizens were in their reports about how the migrant group behaved. And for our results, before we want to get into the metacognitive results, I wanted to point out that the imageability condition and manipulation had no effects on the likelihood ratings of how the group would behave during the extrapolation tasks. So both conditions rated the negative traits as equally likely to occur and the positive traits as relatively unlikely to occur. In terms of the metacognitive results, the low imageability condition reported that they knew more about how the migrant group would behave compared to the high imageability and rehearsal conditions. They were also more confident that they knew how the migrant groups would behave compared to the high imageability and rehearsal conditions. And lastly, they reported more accuracy in the initial information that was perceived about this group compared to the high imageability and rehearsal conditions. And this is really interesting considering that all participants were reminded before the study that they knew nothing about how the migrant group would behave or anything about the group from Ossetia, and each condition had the same amount of information about the group, but the low imageability condition believed that they knew more about the group compared to the other two conditions. So for our discussion, the current results establish an initial link between thinking about different types of trait formation during extrapolation tasks and subsequent cognitive measures. And we think that these effects might be due to the cognitive effort participants in the low imageability condition had to exert to make sense of the traits that were presented to them about the group at the time. And we plan on conducting future studies where we will manipulate cognitive effort in different ways, such as manipulating cognitive load or how easy it is to form a gist of the group information. But we are excited about this research, and I want to thank you all for letting me share with you all today. So thank you.
Okay, thank you so much, Monica. All right, we have a question from Kaylee Decker. How was the trait imaginability determined?
So we had 100 MTRIC participants previously rate an extensive list of traits that were intended to describe groups on a different number of factors, including how likable the group would be if they had the traits and how easy it would be to imagine the groups expressing the traits.
And how did that sort of as a corollary for myself, how did that then get you to the imageability?
So to name an example of like the high imageability and like low imageability traits that we like presented, some examples of the low imageability traits were like ethical, disloyal, trustworthy, resentful, dependable, like just like that. And for the high imageability, we had things that were like relatively more easy. So generous, unfriendly, kind, aggressive, pleasant, things of that sort.
Okay. Do we have any other questions for Monica? Right. Okay, I think you got off.
Okay.
All right.
Awesome. I'll stop sharing my screen.
All right.
Thank you.
All right. Okay, I'm going to give us a 1-minute breather David, could you start sharing your screen? You should be able to
Sure.
set up since we can actually. Okay. All right. Okay. Oh man, you're using the I'm gonna have an issue pronouncing your title. I'm sorry.
No worries. You can just say David.
All right. So our next speaker is David Ferrier. Did I get that even close?
Yeah, that's all right.
All right. He's going to be telling us about hydrogen peroxide disproportionation with manganese macrocyclic complexes of cyclin and piclin.
Yes.
All right.
All right. Thank you very much to attend my presentation. And as Dr. Bovil mentioned, I'm going to be talking about the activity of hydrogen peroxide disproportionation, but some of our complexes that we synthesize in Dr. Green's lab here at TCU. And for this project, I want to talk to you a little bit first about the enzyme catalase, which is the one that inspired this project. Catalase is an enzyme that balances reactive oxygen species. It's found in living organisms exposed to oxygen. Like I mentioned, catalyzes the decomposition of hydrogen peroxide to less toxic species like water and oxygen. And by doing so, it protects the cell from oxidative damage by reactive oxygen species. But the characteristic of catalase that interested for us the most as inorganic chemists is the fact that it contains a metal center. And this metal center can be either iron or manganese. For iron, it's observed as iron protoporphyrin prosthetic groups. And for manganese, it's found as these binuclear species or dimers. So in our group, we synthesize a few complexes with manganese. And today I'm going to talk to you about the complex of cyclin and the complex of piclin, which were the ligands. And we observed that they form dimers as well. So these caught our attention. We decided to use a spectroscopic technique called electron paramagnetic resonance. And we observed that the results resemble the ones of the catalase found in nature. So in the top, we can see the results, for example, what we see in the spectra for the catalase. And in the bottom, we see the one observed for our piclin manganese complex. So it resembles the same, which gave us a hint that maybe our complexes had catalase activity or exhibit this activity. So for doing so, we designed a little setup on how to do that. So our reaction is converting hydrogen peroxide into oxygen and water. So we wanted to quantify the amount of oxygen produced by our catalyst. So I had a pressurized cells right here, which is sealed. Our complex is in solution right here in the bottom. And I completely sealed this, this vessel and pushed all the oxygen with nitrogen. I also have a sensor inserted there, which is an electrochemical unisense sensor specifically for oxygen. And then I characterize my baseline before I add hydrogen peroxide. After a certain amount of time, I inject hydrogen peroxide and then oxygen start to form and I can quantify that. And one of the first things that we observe is like immediately after the injection of hydrogen peroxide, we have this green color in solution and we can start seeing some of the bubbles that it forms since it's oxygen as part of our products. And then after 30 seconds, we have a lot more of it, but the solution turns clear. So we were interested in seeing what was happening here, what was the species that was active for the disproportionation of hydrogen peroxide. So in collaboration with Dr. Sherman here at TCU, we observed that these compounds are actually in, when they're in solution, there's an equilibrium between the monomer and the dimer. But here, when we add a full equivalent, so each triangle is half equivalent of hydrogen peroxide, but when we inject a full equivalent of hydrogen peroxide, we observed the formation of this green color. And this green color was captured at an absorbance of 554 nanometers. And after that, the color disappears. So this color and this specific absorbance is related with dimers of manganese. So we were able to observe that the dimer was the active species toward this proportionation of hydrogen peroxide. We also look at how their structure of these ligands affect their activity. So like I mentioned, we had piclan, which piclan contain this extra pyridine ring, and cyclen, which doesn't contain that pyridine ring. And we observed that the extra rigidity that the pyridine ring give to the structure also helped in increasing the reactivity, or we observe way more reactivity with pyridine, with piclan complexes of manganese. Here I plot proton partial pressure of oxygen versus time, so we can see how we get more oxygen in the presence of piclan. We also did this study in the presence of different pH conditions, and we observed that at pH 8, we have the higher activity, which is concomitant with the fact that at pH 8, we have the higher speciation for the complex of manganese. So with this work, we have some future directions for that, which I'm now working with a bigger library of ligands and looking at how different electrodonating groups and addition of extra pyridine rings affect the reactivity of this complex, and also doing some computational studies to understand the mechanism. With this, I would like to acknowledge Dr. Kayla Green for her help and receiving me in her lab, and Dr. Benjamin Sherman and Deborah Berry, which were collaborators in this project, and TCU for giving me the platform for working on this.
Thank you very much, David. All right, does anyone have any questions for David? So what are the sort of real world, sort of longer term applications for this?
Yeah, so this is a fundamental study, but of course there's real applications in mind. So one thing is like, there's not really an understanding right now of how catalase actually works in nature. We don't really know that. We have some hints in the literature. There's has been a lot of work into it, but there's still some work into finding a small molecules that can resemble the activity. So we can actually figure out how specifically is the mechanism of how it makes oxygen and water. It's also important because some people have deficiency of catalase in their bodies or other enzymes that help them with reactive oxygen species. So small molecules could potentially in the future be used as therapeutics for these kind of health issues.
Okay, do we have any other questions? Okay, awesome job, David. And thank you so much for
Thank
your
you.
Okay, all right. Rachel, could you share your screen for us?
Are we waiting a couple minutes?
Yeah, I think we're gonna wait. I don't want to get too I want to try to stay as close to on time as possible. So if you want to turn your camera off for the 1 minute that we're all going to be staring at you.
Right.
All right, are you good to start? So I'll just do a quick introduction. So our next presenter is Rachel Aines Thaluber, who is going to be talking to us about a question of where differential survival, growth rates, and diets of captive bred hatchling Texas horned lizards reintroduced at separate locations in Mason County, Texas.
All right, so thank you everyone for being here. So my research, if it will go forward. Okay, my research focuses on Texas horned lizards, which are often characterized as dietary specialists because the adults typically eat something like 60 to 100% Pogonomyrmex, which is also called harvester ants. Unfortunately, being so specialized means that they're really vulnerable to habitat alterations. And what we've seen is that their populations have declined very significantly in the last 50 to 75 years. In an effort to kind of combat this, there have been a lot of efforts to breed this animal in captivity and reintroduce it to the wild. But one of the kind of obstacles of this is that most reintroduction efforts use hatchlings, which are less than 6 months old. And we know very little about the habitat requirements for lizards this young, which makes it really hard to pick suitable reintroduction sites and could negatively affect reintroduction success. So the goal of my study was really to try and determine whether location influences the short-term reintroduction success of captive bred hatchling Texas horned lizards. It's a mouthful. But I wanted to look at 3 things specifically, looking at their diets, their growth rates, and their survival to hibernation. To do this, I studied over 250 hatchlings bred by the Fort Worth and Dallas zoos. They were divided into 2 groups and released at 2 locations roughly 50 meters apart at Mason Mountain Wildlife Management Area in Central Texas. And this all occurred last fall in 2020. Then what I did was I tracked them for the following 3 months until they reached hibernation. And how I did that was the lizards had these little tags glued to their back. And what that would do is it would cause a receiver to beep when I got close. So I could basically find them about 3 to 4 times a week, kind of mark where they were and what they were doing. Once a week, I would capture them and weigh and take a length measurement. It's called snout to vent length, so the length from the tip of their nose to their cloaca. I also opportunistically collected horned lizard scat, which you see on the right of the slide, which I then dissected and identified insect fragments to assess their diet, which is a method I've found works very well for looking at the diets of these lizards. So when we looked at the diet, we saw some pretty substantial differences between our 2 release sites. So at what I'll call site 1, the lizards ate almost exclusively an ant called chromatogaster ants, which is shown in blue. Whereas at our second release site, they ate a much more diverse diet of a wider variety of ants, as well as a lot of termites. Now, in itself, a more diverse diet is not necessarily a bad thing. But what we also saw was that lizards at site 1 were eating more prey per sample. So basically they were eating more food than the lizards at site 2, which suggests it's not necessarily a great diet at site 2. When we looked at how much the lizards were growing, when it came to length, there weren't any differences between our 2 release sites, although our second site, there was much more variation in how much their length increased. However, there were significant differences in weight gain. So lizards at site 1, where they're eating more, on average put on more weight than the lizards at site 2 did. When it came to survival, I categorized the lizards into 4 groups. Lizards that were dead are shown in gray. The percent that were predated is shown in orange, which basically means I found their tag in fecal material of a predator. Lizards that we don't know what happened, that's shown in yellow, and that could be that they shed and I haven't found them since then, or possibly they got predated, but I never recovered their tag. But really what I want you to focus on here is the blue section, which shows the percentage of lizards that were alive at each site at hibernation in December. And there were significantly more lizards still alive at site 1 by the time of hibernation. When we think about kind of interpreting these results, it's important to remember the home ranges of these lizards are very small, like 1 to 3 square meters maybe. So even though they're roughly 50 meters apart, it's still, it might as well be on opposite sides of the planet for these lizards because they're not moving very far. And as far as we can tell, it seems like site 1 is a better location because they were eating more food, they were putting on more weight, and they had higher survivorship. What is the underlying cause of this? We're not entirely sure. It could be that lizards at site 2 have a suboptimal diet. We know that the chromatogaster ants that they were not eating at that location are an important food resource. So the fact they're not eating those could explain why they're not gaining as much weight. And this could be a reflection of differences in prey availability. Maybe those ants just aren't as abundant at site 2 as they are at site 1. Some other things that could be coming into play though are vegetation and soil hardness, which can affect basically how the lizards can avoid predators or thermoregulate. But overall, it seems like location is probably an important part of reintroduction success for hatchling horned lizards. So in the future, I'm going to be looking specifically at how the prey availability, vegetation, and soil hardness differ between those 2 locations to figure out why. There's a lot of people that have collaborated to help with this research. I cannot take the time to thank all of them, but I greatly appreciate everything they have all done. I will take questions.
All right. Thank you very much. Okay, does anyone have questions for Rachel? Okay, I actually have a question from our awesome tech support. If just a 50-meter difference had just a drastic effect, how can you survey a larger area?
I don't survey a much larger area. Like that, they hang out in a pretty small area and I just kind of go where I know that they are and check on them. On occasion, I go further out to see if they have dispersed, because occasionally they do that. But really they tend to hang out in a pretty small area. Makes my job easier.
So I actually have a question. So you mentioned sort of looking at the soil. How are you going to go about doing that? Sort of like what tests?
I think we're going to get like a penetrometer, because the biggest thing is, can the lizard bury in the soil easily? So how hard the soil is, I think getting a penetrometer would work pretty well for that.
Okay. All right. We have time for one more question for Rachel, if there are any takers. Okay, thank you so much. All right. All right, Davis, could you share your screen for us?
Yes. Okay.
All right.
Good to go?
All right, yeah, I'll do a quick introduction and you're good to go. All right, we're next going to hear from Davis Martin, who's going to talk to us about assessing the effects of inherited mutations on PALB2 structure and function.
Okay, thank you. So my research really centers around this protein, PALB2, which is an incredibly important protein within the field of breast cancer research. The reason for this is that along with BRCA1 and BRCA2, these 3 proteins are incredibly important for repairing DNA damage, and thus they have a strong tumor suppressor function. The way this occurs is that when damage occurs in the DNA within the cell, these proteins become activated and they bind to each other, forming this heterotrimer complex that's capable of binding to the site of damage and repairing it. And this effectively stops tumor formation. However, some individuals, especially those who have a family history of breast cancer, often inherit mutations within the PALB2 protein that causes a loss of binding to BRCA1. And this causes BRCA1 alone to be recruited to a site of damage, and it's unable to repair it. What this does is it leaves the person vulnerable to basically accumulate damage throughout their lifetime. And this accumulation is what eventually can drive tumor formation. However, not all mutations within PALB2 are equivalent. It needs to be experimentally determined whether or not these mutations might be cancerous. And the way we do this is by looking at the structure of the protein. So it has 2 main important functional domains: the WD40 domain where BRCA2 binds, and the coiled coil domain where BRCA1 binds. Mutations within these domains have a much higher chance of being detrimental to the protein's function and eventually cancerous, whereas mutations outside of these regions are most likely benign. However, experimental testing is needed, and so we call all these mutations variants of unknown significance. With that backstory in mind, our project had 2 goals. The first was to test a new in vitro method for determining the effects of these variants of unknown significance on PALB2's function. And if the method was successful, we then wanted to attempt to classify some of these variants as either detrimental or benign. So to start the project, we don't know the exact binding interface in the coiled domain that BRCA1 uses, but we do know that PALB2 binds to itself in a known coiled structure, and we hypothesize that given the large overlap in this binding domain, that BRCA1 probably uses some, if not all, of these same amino acids for its binding interaction. Going off of that hypothesis, we analyzed a database of mutations that have been inherited by patients with a family history of breast cancer, and we selected 3 mutations to make: L35P, K30N, and L24F, where the mutations are labeled such that this represents the amino acid present, 35 is the position, and then P stands for proline, which is the amino acid we mutated it to. We hypothesized that L35P and L24F would have the greatest detriment to the protein's function because it's thought to be in the binding interaction. And K30N we thought to be rather benign since it's thought to face away from the interaction. We then measured the effects using isothermal titration calorimetry, or ITC. And that was the first goal, was to try to show that this is a good method for doing this. And the way ITC works, very simply, is that BRCA1 is in a syringe and it's slowly injected or titrated into PALB2. And the changes in temperature from the binding interaction are actually measured using an internal thermometer. And those changes are then graphed over time and fit to a binding curve so that we can create a binding constant for each of our constructs. So at the end of an ITC experiment, you see that all of the BRCA1 has been injected into PALB2 and it's found in a bound state. Now, when we get the data and we fit those, the curves, we get a quantification for each construct. And when we got that for our data, we see here, this is our wild type or unmutated PALB2 protein, and then each of our 3 mutations. And graphed on the y-axis is the quantification of the binding affinity. So how strongly did these proteins bind to each other? And it's an interesting concept where the smaller the number, actually the greater the affinity. So we see that wild type PALB2 had the greatest affinity. L35P had no data because we saw no binding upon mutation. So it had a serious effect on binding. L24F lost about 100-fold binding affinity, and K30N lost about 10-fold, although the error was quite large due to an error with fitting the data properly. So the data was reproducible, but the fit curve was inaccurate. So further optimization should help us to decrease this error. And overall, our data showed that ITC shows good promise for using it as a method for determining the effects of mutations on PALB2's function. And in the future, we hope to create more mutations to further optimize the process, as well as provide more helpful information for knowing when are these mutations detrimental and might require some preventative measures to be taken in patients who have them, or if they're benign and nothing needs to be done. So thank you, and I'll take some questions.
All right, we have time for one question. All right, I will then take host prerogative and say what steps are going to be required before this could actually be sort of used practically.
So the main thing is that no one experiment can realistically determine the effects of these mutations. So a main goal will really be, we have a lot of in vivo data, but that needs to be paired with the structural data so that we understand the why behind why some of these mutations seem to be detrimental. And I think the accumulation of all of the data together can create a comprehensive profile of each variant so that we have the best information moving forward.
Okay. All right. Any other questions? Okay, thank you so much.
Thank you.
All right. Jacob, could you share your screen for us?
Absolutely. All righty.
All right, we're gonna wait a second.
Just a second.
We're running about a minute ahead, and so I don't want to. I want to start you right at 1:58.
Perfect.
Just in case you have any last minute supporters popping in.
Got it. Sounds good.
Okay. All right. So our next and final speaker for this session is Jacob James, who's going to be discussing use of DNA barcoding to distinguish between morphologically similar red bats. All right.
All right. Thank you so much. Howdy, everyone. My name is Jacob James, and today I will be discussing my research project on using DNA barcoding to distinguish between morphologically similar red bats. I want to give a thank you to the Science and Engineering Research Center and the CERC grant we received to do this project, as well as Dr. Hale and Dr. Williams for all their mentorship and all of the help they were to me in doing this project. So first off, the basis of this research is to determine between two similar species of red bats, the eastern and western red bat, with the hope of developing conservation strategies for them when we look at wind energy facilities in the United States of America. So the first of these species is the eastern red bat. It is a migratory tree roosting species, which means it migrates and lives in trees. As you can kind of determine from the eastern name, it inhabitates eastern North America, primarily that region that's highlighted on that map, as well as the South and Central Texas, North and South Central Texas region, where most of our research has been conducted. The next species is the western red bat, which originally we thought was a subspecies of the eastern red bat, but with some recent DNA technology and discoveries, we've actually understood that they are two distinct species of bat. It normally inhabitates it lives in western North America, as you could probably suggest or understand from the name, as well as down to South America as well. The big key for the western red bat is our normal habitat distribution that we see with them does not include North and South Central Texas, where most of this research was conducted. However, I will get into this in a minute, that we actually do have reason to believe that they have begun inhabiting this area and have actually expanded their habitat, which is important for our research as a whole. So looking at wind energy in the United States, currently it is the largest source of renewable energy in the US. And in fact, Texas actually has the most installed wind energy capacity, which simply means that Texas produces the most energy in the United States from wind. As you can see this pretty graph on the right, it's really easy to follow that the wind energy has increased a lot over the past 16 years or so, which is important for our nation as a whole, but also important for us as researchers to understand what is happening to these species, especially these migrating bat species at these wind energy facilities as they continue to pop up. And as you can kind of as you could probably suggest by what the point of this research is, they are being killed at these wind energy facilities. We have some theories why, and we have some theories how we can stop that. One of those theories is the ultrasonic acoustic deterrent, or UAD. So normally how bats navigate throughout the airway is they use ultrasonic frequencies, much like a sonar. They beam out ultrasonic frequencies, it bounces off something, they interpret that signal, and they navigate as a result. The idea of a UAD is to also send out a bunch of ultrasonic waves and make the airway kind of noisy for them. And whenever they find a cloudy, noisy airway that they can't effectively navigate, they end up just going around it. Or this is at least what we've observed in certain studies. So the idea here is to prevent them from dying at these wind energy facilities by flying into the wind energy facility, by just having them go around by using a UAD. And that's kind of what previous research has shown to be effective. First off, however, I do want to hit on, last year there was a study that came out about the diversity of bat species in the South Texas region, mainly the Rio Grande Valley. And this was the first time that we saw L. blossavelli, or the western red bat, in these regions that we have never observed them before. And that's why this is important, is because if these western red bats are now expanding their habitat into Texas, where the most wind energy facility is, or the most installed wind energy is, then it's important for us to be able to know that, and thus calibrate the UADs effectively in that way. Also, previous research has shown that UADs have been effective for 2 other species of bat, the Mexican free-tailed bat and the hoary bat. This is important because they echolocate at different frequencies than the Western and Eastern red bats. So if Western and Eastern red bats also require UADs, we know that we will need to calibrate them differently. And that's what our current research is focused on. This is in collaboration with Texas State University, being conducted in Hays County, which is South Central Texas. Basically, this flight cage experiment is where you take bats from the area, which is high in bat biodiversity, put them in this flight cage and see how they respond to a UAD. See if they fly around it, see which frequency works what best for them, so on and so forth. And that's the point of my research, which was to determine Western and Eastern red bats that are being involved in these flight cage experiments to see if UADs are effective for them or not. Like I said, we've never seen Westerns before in this region, so we kind of thought everything we were testing that looked like an Eastern red bat was an Eastern red bat, but we need more genetic testing to kind of figure that out. So the methods, Christmas came early for me this year, I guess. I got 19 fecal samples from the bats in Hays County. I was able to extract the DNA and amplify it at a 120 base pair section of the cytochrome oxidase 1 region, which is the barcoding region. That's just important. It's mitochondrial DNA, but it's important because whenever we put it against the database, that's the region that can distinguish between the Eastern and Western red bats. Put it in a genetic analyzer, and we're still waiting on the results. They're on the horizon, but we do expect to see a difference between Western and Eastern red bats. We do expect to see both species that are being involved in these flight cage experiments. And whenever we do get that data, and if that is what it shows, we know that we will need to re-examine past data on UADs for species-specific differences and also calibrate UADs differently in the future. So overall, we do know that UADs are effective for the Mexican free-tailed bat and the hoary bat. We just need to make sure that it's also going to be that they're also going to work for Eastern and Western red bats. This goes to show the importance of collaboration between different entities, the governing entities of the United States, wind energy developers, Texas scientists, Texas Park and Wildlife, just about everybody, because we do want to see more renewable energy in the United States of America. And wind energy is a great solution to that, but we don't want to be putting these other species at risk while we move towards that. So that is all I have. Thank you.
Okay, we have time for one quick question. All right. How do you physically distinguish an Eastern red bat from a Western red bat? Can't you just look at them or look at the dead ones near the base of the turbine?
So that is
Shager.
Yeah. So you can somewhat field identify, and that's actually been the basis of past research. And that's why that study from last year that genetically showed differences in the Western and Eastern red bats and found genetic differences or like genetically discovered that Western red bats were in that region. Normally, Western red bats will have dark colored ventral fur and Eastern will have light colored ventral fur. But we kind of learned last year with that research that it doesn't always correlate. It's not always like a one-to-one. It doesn't always work like that. So in the field, we can have a pretty good idea of what the species is based on how it looks, but the genetic testing is actually what confirms it whenever we look at the DNA.
Thank you very much. And that ends this session.